What?
Want to go to DC?
Oh, okay. And forever, what's the one you guys want that you went to? You say like your grandma's one?
Yeah, that's cool.
Anybody need the handout from last time? Anybody need a 2022 Brown Swiss calendar? What? Anybody need it? It's up here. Came in the mail.
Lost my question thing. I'm not
Okay.
sure later.
Okay. There's, you can do that, or there's also blank ones on the inside there. What you've done is great, but if you want some for the future, there's extras on the inside. The forms,
Yeah. Where? Inside of
they're
where?
inside the jug here.
Oh. Got it. Thank you.
There you
Yes.
go.
Sweet.
Yeah. Everybody have assignment 6 and today's handout.
I don't have it.
What happens when the squirrel comes along and gets it back? Anybody else? Assignment 5, we got any more up here? Turn that in.
I thought so.
Take a look for it again.
Oh, yeah, I did it.
Okay. I just didn't see it in the file. Okay, I think we got everything handed out. So review session after class today. If you have questions about the homeworks, the assignments, that's a good time to ask those questions. I got assignment 5 from everybody. 6 is up on Blackboard. You got a hard copy of that. You got your next question due. I'm assuming most of you put that in the jug. We'll probably need to get those answered before Tuesday, so I'll try to get the answers posted up on Blackboard. First exam is next Tuesday. Coverage 1 through 9, all the way up to the end of feet. We don't go into the bones. And basically the assignments 1 through 4. So I tried to get those back to you today. And then 14th, next week from today, we're going to meet up at the other classroom and do some hands-on stuff with muscles and bones and the heart, things like that. So after break, I've been told they're going to renovate this part of the building. So stay tuned for wherever we have to end up. I don't know where they're going to assign a room to us. So, yes.
Did you take homework?
I sort of, yeah. You have homework? Okay. Anybody else have the assignment 5?
Open during this time, or is there a classroom?
I don't even know. I think it's open. There's a lab at 10, maybe. It may bleed into this hour. I'll have to check with Professor Marshall, but we won't be spending a lot of time there. We'll move where we need to. Grading assignments, sort of where we go with those. Remember, in this class, we're going lecture, determines the assignments, and then I use the assignments to determine the exam. So if you can answer the questions well on the assignment, you should be able to do well on the exam. Questions or concerns? Okay, letters. Let's see if we've got a couple things figured out here. Which increased blood calcium or put blood calcium into the exchangeable pool? So osteocyte is what we've been focusing on, so D, but osteoblast will as well. Which increased blood calcium or put blood calcium into the exchangeable pool? Increased PTH, increased calcitonin, increased vitamin D, decreased vitamin D. So certainly PTH, A is going to get it done. C will also help as it promotes absorption from the gut. The exchangeable pool of calcium in a cow is about how much? 1 gram, 10 grams, 100 grams, 1,000 grams. So the exchangeable pool is B. We're looking at the amount of calcium in a bottle, calcium gluconate, that's going to fill up the blood again, fill up the muscles again. So 10 grams. 10 grams of calcium in a bottle of calcium is about equal to what? The amount of calcium in the blood and muscle, the blood and the bones, the muscle and the bones, or the muscle and the gut. So 10 grams, we're looking at the exchangeable pool, which is blood and muscle, A. Dry cow, the calcium in her feed flows to where? Calcium she consumes ends up where? The manure, the fetus, the mammary gland, or the bones. B is going to be our focus, but you could also say that it ends up in the manure, ends up in the bone. And those are correct answers, but not as strong as the fetus. Largest calcium demand, the lactated dairy cow, is from what? The gut, the fetus, the mammary gland, or the bones? So the mammary gland, that's our biggest export, the biggest draw, 1 gram an hour. Subclinical hypocalcemia in our transition cows stops skeletal muscle contraction, lowers feed intake, increases uterine contraction. B, stops smooth muscle contraction, increases feed intake, increases uterine contraction. C, increases skeletal muscle contraction, lowers feed intake, lowers uterine contraction. D, stops smooth muscle contraction, lowers feed intake, and lowers uterine contraction. D is what we're looking for. The stoppage of that smooth muscle is going to have those downstream consequences of lower feed intake and lower uterine contractions. So last time we started muscle types, discussion of muscle, 3 types of muscle, categories, skeletal, cardiac, and smooth. You can put them in any order you want. Skeletal, they are that striated or banded appearance we'll talk a lot about today, and they're voluntary. We have conscious thought about how those muscles get moved. The cardiac and smooth are involuntary. We don't have to think about them contracting. Cardiac tends to have a more striated appearance, and our smooth muscles are not organized in such a way that we get that striated appearance. So moving on to new, you should have this handout from last time to fill in this table. So the approximate composition of mammalian muscle, the component, the percentage, and the range. What are we looking at for amounts? So water is going to be a big component of our muscle tissue. 3/4 of our muscle is water. 65 to 80, probably depending on whether or not we've gotten a lot of extra fat floating around. So anybody here make their own jerky? What happens to that as it dries out? Shrivels up considerably, and that's that loss of water. So it's like 1/4 of the size it started out with. We lose that water. I don't like to lose all of it. I like a little bit of chew to my jerky, but that's a personal thing. So the other major component we're looking at is protein. We'll talk about what those proteins are today. We're looking at the volume is contractile proteins, and you'll see that in our diagrams. But you have all the other proteins necessary to make the cell go. So all the enzymes, all of the structures, all of the receptors on the cell membrane, the pores on the cell membrane, all made of protein. Lipids, we're looking at 3%. So what kind of lipids do you think might be present, given that range of 1 to 13? So you have muscle, that tissue. What forms of fat are we going to have? Certainly, we're going to have those two types. What about functional groups? In the sense of this fat does this stuff and this fat does this stuff. Phospholipids for the membranes. So all the cell membranes are going to be made of phospholipids. That's going to come into that 3%. As we get more and more triglyceride in the cell, uses energy, that proportion is going to go up. So phospholipids and triglycerides are very important in muscle. Non-protein nitrogenous substances. So what might non-protein nitrogenous substances be? What has nitrogen that isn't protein? What contains a nitrogenous base? DNA and RNA. There are a lot of nucleuses in our cells. So DNA, RNA are going to be that 1.5%. Carbohydrates, 1%. We're looking at glycogen stores. So if we can carb up good the night before an event, carb load, we might get up to that 1.5%. If we're doing a marathon, we jog along and we hit the wall, we've probably dropped that carbohydrate to 0. We've used it all up in the short run. Last part, inorganic constituents. 1%. What's another word for inorganic constituents? Anybody want to hazard a guess? Minerals. So calcium, potassium, maybe a touch of sodium, chloride, all those macro minerals, and if there are any trace minerals as well, but the macro minerals are going to take up the bulk of that 1%. So as we move forward, I think I mentioned this in feet already, but just making sure we have it down. A tendon versus a ligament. A tendon connects bone to muscle. So usually involved in movement. Those tendons slide back and forth and hopefully move things the way they're supposed to. Ligaments are going to hold the bones kind of in place. So they are bone to bone, especially across a joint. So you've got ligaments on all 3 sides, all 4 sides of your knee, all 4 sides of your elbow, hundreds of them in your fingers and stuff, holding those bones in the proper orientation. So both are made of a protein base. Got that connective tissue there. Protein base, you've got some sugars and a couple other compounds involved to keep things saturated, to keep things lubricated and moving. So we're going to talk about muscle fibers today. Muscle fibers is the specialized name for a muscle cell. Instead of calling them muscle cells, they've decided they're going to call them muscle fibers. So a muscle fiber is a muscle cell. If we look at kind of the structure of a muscle, we've got a tendon connecting to the bone. And then we have, in this case, a skeletal muscle where everything's kind of running as an extension of that tendon. The idea is that we have contraction that's going to pull on that tendon and move the bone around. We've got individual muscle bundles moving through, and we get down inside that muscle bundle to a single muscle fiber, a single muscle cell. So number of cells linked together, lying in parallel, that allow that muscle to contract and exert a force on whatever that bone needs to do. So working our way in, we have that individual muscle cell sticking up out of a muscle bundle. So muscle cells long and thin. Inside, they have myofibrils. These are cylinders that make the contractions happen. Those myofibrils are made up of filaments. So we have different proteins, different filaments that are going to come together to make that myofibril. We'll talk about the sarcoplasma, which is the cytoplasm in a muscle cell. The sarcolemma, which is the cell membrane. And we tend to have the nucleuses of this muscle fiber on the outside. And we'll talk about that as we go through. So if you're thinking about muscle organization going from big to small, we have the muscle. The muscle is made up of muscle bundles. So little sections that probably have the same nerve root, individual ends of the same nerve. That muscle bundle is made up of the individual muscle cells, the muscle fibers. Inside our voluntary or skeletal muscle, we have the myofibrils. And those myofibrils, myo meaning muscle, myofibrils are made up of myofilaments. So trying to keep them in order can be a little bit confusing. Well, we focus on muscle, and then myo as we get smaller and smaller. And it appears the words get longer as we get smaller. Might be a Napoleon complex. So muscle fiber, muscle cell is a highly specialized cell, as are most cells in the body. They'll differentiate in different ways and focus on different parts of the DNA to make what needs to happen. So we're thinking about a muscle fiber. Instead of thinking about a cell that tends to be generically round, we're dealing with something that's very, very long, almost long enough to see with the naked eye. So muscle and avian skeletal muscles, long, unbranched, thread-like cells. Get into cardiac, they branch, and they taper down to a point at either end. So long, thin, with a point on either end. So these fibers, I said, an individual one, maybe several centimeters long, and that we start to see that maybe we could see that, at least that thread look end to end with the naked eye, but they rarely extend the entire length of the muscle. You tend to have everything over like bricks in a building, everything kind of coming together and then working as a unit. And there are probably some advantages in not going the full length, but I think we can get the muscle a little bit shorter if it's not just one cell. So picture we showed you before, long tapered cells, points at the end. So each of those is an individual cell that we're seeing sliced through. Does anybody want to guess what all those little black dots are? Black dots are the nuclei associated with each of the muscle fibers. In all cases, we have multiple nuclei associated with one muscle fiber, which is a unique feature to these particular cells. You see it in white blood cells, but not everywhere in the body. So how does this structure differ from the generic animal cell that we put together before? What might some of the visual differences be between these two cells? So we've got the nucleus inside versus out on the periphery. Long and tapered, round. You're not seeing a wide range of things, at least readily apparent. There's a whole lot happening in here, but this looks pretty uniform. And that comes back to the cells are dominated by those long myofibrils that run the length of the cell, the contractile component. Everything else gets pushed out to the edges. We need uninterrupted motion there. So in the same muscle, the diameter of the fibers vary greatly. Has to do with how much those muscles used, how much training you do. You can affect the number of myofibrils in your body and the thickness of those, but you can't change the amount of muscle fibers. Muscle fibers, muscle cells are set as your body develops, so they're set by birth, probably long before that. So the diameter, we're talking centimeters long, but 10 to 100 micrometers in diameter. Very long, very thin. Everybody know what a micrometer is? The 1,000th, 1 micron is 1,000th of a millimeter. So 100 microns is 1/10 of a millimeter. So a millimeter is pretty small. 1/10 of that is the diameter of our cells to 1/10 to 1/100th of a millimeter in diameter. So for whatever reason, they've come up with specialized names for your muscle parts, muscle fiber parts. I'm not sure if I gave you this one or not. I hope I wanted to. I don't know if it got printed out. No, it would have been from the other day. I'm guessing I missed that when I printed the other one. I forgot to do 2 of them. Very, very small.
You put the approximate dimensions of them. They have a chart of that.
Okay.
Or,
So you don't have this table ready to be filled out. So looking at the muscle fiber parts, everything it seems related to a muscle fiber, a muscle cell, has a specialized name. So instead of the plasma membrane, we're calling it the sarcolemma. So that's the membrane surrounding the muscle fiber, i.e. the plasma membrane. One of the unique features about it, it's got a little bit more protein in it to make it more elastic. So as the fiber contracts, the sarcolemma contracts with it. As it relaxes, it expands back out again. So the sarcolemma is that membrane. It comes from the Greek meaning husk. So you've got this membrane around the outside, maybe a little bit tougher than our typical cell membrane, allowing that cell to change lengths and that membrane to adjust accordingly. So from the Greek words, sarco meaning flesh, lemma meaning husk. So sarcolemma. So your diagram, this kind of grayish purplish outside is our sarcolemma. A little bit more elastic than your typical membrane. So we've got that diagram, and then just a little bit darker. I assume these are sliced, frozen and sliced, so you're just seeing that little bit of darkness on the outside edge. That's the sarcolemma as we lay those things out. Sarcoplasma, basically it's a cell contents, basically it's the cytoplasma or cytosol. And as you might guess, given the need, there are a lot of mitochondria in our muscle cells. We need those mitochondria to generate those ATP. The ATP are going to run the muscle contractions, so we need a lot of them in there to get that done. And depending on our diagram, you'll see those or not see those, but they're all over inside the cell. So mitochondria, they're going to be dispersed in between the myofibrils. They'll generate the AP, the ATP slides out to the myofibrils and provides the energy for those myofibrils to contract. Nuclei. So the nuclei, muscle fibers are multinucleated. An individual myofiber could have hundreds of nuclei associated with it. You can sort of see the start of that in that one picture I gave you. And they're all pushed to the outer edge, pushed out to the sarcolemma. They're not in the center of these myofibrils. So those nuclei in this diagram, we're seeing the bumps on the outside. Looking at that diagram, why do we sort of need them out of the center and push to the edge? Not enough room, or they're going to interfere with those myofibrils. The myofibrils have the contractile part of the cell need to run the entire length of the cell. If we put a nucleus in the middle, that's going to mess things up. So those nuclei get pushed to that outer edge. All those little black dots, generally on the outer edges in our diagram. Then we have the transverse tubules. This is an area, or one of the areas, that store our calcium. We also have the sarcoplasmic reticulum. Transverse tubules, the T-tubules, or the sarcoplasmic reticulum are going to store our calcium. We can think of them as constantly trying to get the calcium back into the inside. If they're stimulated by a nerve, they release the calcium into the cytoplasm. The cytoplasm starts the chain reaction, the system of chemical steps that lead to muscle contraction. So as long as the calcium is in the T-tubules, as long as the calcium is in the sarcoplasmic reticulum, there is no contraction. Or if we have no calcium in the muscles because of no fever, we have no contraction either. So what we're looking here, transverse, they work their way all the way across. You have the transverse tubules, sarcoplasmic reticulum, working together to hold all that calcium. We have those storage boxes for calcium. They surround the microfibril. They work their way across the cell. Everywhere there's a myofibril, we have them surrounded by those two components. Each myofibril has one connection to the nervous system. That connection is called the myo for muscle, neuro for the nervous, so myoneural junction. So every muscle fiber has its own nerve. This is why we have kind of our muscle fiber, they're kind of set at birth. We can't be adding and subtracting muscle fibers, and then what do we do with the nerves that are just hanging there? We have to grow new nerves. So what we're doing with the muscle fiber through training, through growing up, through getting older, is making that individual myofiber stronger, bigger. We're going to increase the amount of tractile components, the myofibrils, in each of the cells, but the cell itself is still there. So I don't think there's a neural junction in this picture. There's other pictures we have moving forward, but there's a flat spot on each of the nerves where each of the nerves make contact with that sarcolemma. The sarcolemma sends that signal to the T junctions, or T-tubules, sarcoplasmic reticulum, and that releases the calcium into the sarcoplasm. The last part is the unique feature to muscles. We have the myofibrils. They are the long cylinders that run the length of each cell. So contractile protein cylinders, and what we'll have is a repeating unit called a sarcomere in our skeletal muscle. So we have chains of sarcomeres together that form the entire myofibril. How we doing? Everybody caught up? So we got a cleaner version that you'll get in your slide set. We look at that myofibrils in the cell. They run the entire length of the cell. In this one, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 myofibrils. I guess 10, 11 myofibrils. We have each of those myofibrils is stacks of contractile units. So we have what's called the Z line, and we'll get into that detail, running to Z line. This here is one contractile unit, and they're stacked on top of each other, going through, and you've got them all linked together. If they all contract, that myofibril will shrink considerably and hopefully allow enough force for your bones to be moved where they need to be moved. So another drawing showing that hierarchy: muscle bundle, individual muscle fibers. Inside there we have the myofibrils, which are made up of individual myofilaments. So again, going back to that diagram, I think we covered all the parts in there. We haven't got striations yet, but we'll do that. So the myofibrils, those contractile proteins, they're unique to muscle tissue. Every cell has the genes for them, but they're only expressed in the muscle cells. Those myofibril cylinders will run the entire length of the cell and be 1 to 2 microns in diameter. How big did we say the cell was? They're 2, 10, 100. So they're going to be 1/50th or 10% or 20%. A muscle cell that's 50 microns in diameter, an average diameter, has at least 1,000 and can have more than 2,000 myofibrils. So the drawing we had, definitely not as populated with myofibrils as what we'll find in life. The myofibrils are composed of two kind of opposing or two different groups of proteins. You're going to have the myosin complex, made up of a number of different proteins. You'll also be called the thick filaments, and we'll show you, that'll become pretty obvious here in a second of why they're called that. So there's a whole bunch of proteins that come together to form the myosin complex. That's the part that ratchets, that moves to make that contraction occur. Then you have the actin helix, which is a number of different proteins strung together in a double helix. And then you've got troponin and tropomyosin attached to those. This becomes our attachment point for the myosin group. This becomes the ladder which myosin climbs up. So the myofilaments are organized into a structure that allows this contraction to occur. The myosin and actin are put together in what are called sarcomeres. This is our smallest contractile unit. And with our muscle cells, they're going to be a block or a section of that myofibril, and they're going to be hooked together like a train from one end of that myofibril to the other. So the actin and myosin in this structure are going to slide past each other, and that's how contraction or shortening of the myofibril occurs. So this is an illustration of that. From here to here is one sarcomere. We talked about thick filaments and thin filaments. This is our thin filament here, this darker black. That's our myosin complex. Then we have our helix here is our thin filament. That is our actin complex. And what happens with those fibers as they move back and forth, they're in the relaxed state, they move together into the contracted state. So what's happening when we look at that? So those two end lines are coming closer together. Do the thin filaments and the thick filaments, the myosin and the actin, do they change in length? No, they're just sliding past each other, just like my fingers. My wrists are getting closer together, but my fingers are not changing in length. So We think about our sarcomeres. They're about 3 micrometers long. The diameter changes with training. So the more contractile proteins there are, the thicker that myofibril gets. Everything's stacked next to each other. They're still going to move back and forth the same way. Within that sarcomere, we have our myosin, about 1.5 micrometers long, actin about 1. So you have an actin coming in from one side, an actin coming in from the other side. That's going to cover about 2 of the 3 micrometers in length. And then we have the myosin overlapping, 1/4 on each side. And it's that overlap that allows that contraction to start moving and bringing those opposite sides together. So the sarcomere, if we're looking at it as a stained tissue, you've got a whole bunch of thick filaments in the center. The thin filaments are coming in from what are called the Z line or the Z disc. The Z line extends the entire diameter, transverses the myofibrils. We have the actin coming in, thick filaments in between. And you can see that we've got a lot of overlap here. That becomes this darker band right there. That overlap here becomes that darker band there. And then we have a protein in the middle that holds those myosin kind of in place. And that forms that center line darker area. Those thin filaments are not overlapped here out on the edge. So we get a lighter looking area. So this is how our striations are formed to the visual eye. Any questions so far? Is everybody following that so far? So we'll start again at 1 of, take your break, and we'll get back to how sarcomeres are set up and organized. This one? Yeah. Okay. You're welcome. So lots of different representations of things out there in the world. This is one cartoon. Here's another cartoon of the same thing, showing how we have in a relaxed state, a little bit of overlap. As we contract, we bring those outside edges, the Z disc or the Z line, together. Basically, the myofilaments, the thick fibers, are climbing into, pulling themselves into those actin helix spaces. So on those thick filaments, you're going to see little bumps. Those bumps spiral around, and the calcium entering allows them to contract and pull. And the attachment spaces on the thin filaments or the actin to open up. So another representation of the same thing. We've got the sarcomere running from Z disc to Z disc. A little bit more illustrative of how those things are shaped. So again, you've got the filaments staying the same length, but as one climbs up or climbs past the other one, it affects the opportunity to pull those Z discs together. So looking at the parts of a sarcomere, we have one running here from Z line or Z disc to Z disc. And then we've got a whole bunch of labels there showing what changes and what doesn't. So this is meant to more look like it does under a microscope. And then this is a stylized drawing to show you the elements as they move past each other. So the thick filaments of the myosin or the myosin complex, they are going to constitute as what is called the A band of the sarcomere. Where we only have thin filaments, we'll call that the I band. Where there's overlap, that is not the I band, that's the A band. So how do we look at that? In our drawing, got our A band runs from one end of the thick filament to the other. The A band never changes in length. That is constant. The I band runs from the end of the myosin filament in one sarcomere to the next sarcomere and the end of those myosin filaments. And as things contract, the I band will be reduced. So as we move together, This area stays constant. These areas, the I band disappear. Well, they get smaller. They're never going to disappear. Don't contract usually that much. So on your other illustration there, the I band starts in one sarcomere, moves on to the next cell, and they're contained together. So in that diagram, that I band area, the continuous I band, comes over into the next sarcomere. The H zone is a middle area of the sarcomere where you're only going to have thick filaments. So that H zone in the center here, that's going to change as the actin or thin filaments move in. The H zone is going to get smaller. Under a microscope, we're going to see these two thicker areas moving together. So the H zone is anywhere there is not thin filaments. The M line is the center of your sarcomere. That is the anchoring point or the stability disc that kind of holds the myosin in relationship to each other. So you've got this area here where you've got a connection between all these thick filaments that kind of holds them centered in the same place. Before and after contraction, we don't have M or myosin areas sliding back and forth going into one and not the other. If something like that were to happen, we didn't have that anchor in the middle, they'd shift one way and then they'd misalign and we'd have all kinds of trouble. The Z line is what defines our sarcomere, so it's the end on either side. M is the center, Z is the outside edges, defines our end. So that Z line is what we see under a microscope, but it's really a Z disc that has the entire cross section of the myofibril. Thought I had a closer picture of that. No, I don't. Any questions on the parts of a sarcomere? So we've kind of looked at everything. We've cut the muscle fiber front to back. Now we're going to go across. So this is kind of a front to back, a sagittal plane that we're going through. What happens if we cut across our sarcomere, making transverse sections? So we take a look at that smaller part of the diagram. This again shows that hierarchy: muscle, muscle bundle, muscle fiber, myofibril, and then we break things down. We get down there. So we have an individual. If we cut through the space here, we've only got thin filaments. Everybody see that? And they're arranged in a hexagon pattern. So 6 actins are going to surround an individual myosin. If we take a slice through just the thick filaments, We can see that they're arranged in that kind of equilateral arrangement. We can see here in the middle, the M line, the interconnection between all those thick filaments that hold them in place. Then we go through here, we're cutting through thin, thick, thin, thick, thin, thick. We can see how they arrange around each other. So you've got one myosin surrounded by 6 actins, and then we have all the little heads of the creepy fingers on the myosin complex that crawls up the actin. So you're looking at your myosin, you've got your different parts here that come together that form that thick filament, the myosin filament. You have the ratcheting heads that go all the way around in kind of a spiral fashion. So they can reach each of these 6 different actins surrounding the myosin. So these little bumps are going to reach out to each of the actins that surround that myosin. Actin molecules, little spheres, connect together, bond together, and then they form that double helix. Inside that helix are the bonding points that these bumps on the myosin filament connect to. So if we're looking at our sarcomere, looking how it's all put together, this is an example of what kind of protein structure. Two or more protein molecules coming together, acting as a unit, that 4th level quaternary structure. So we've got the individual protein molecules, tertiary structure. They come together, attach together to form that quaternary structure. So here's an actual cross section looking at a muscle, so a transverse section. So you can see in this one, we've got the thick filaments surrounded by the actin. Over here, we sort of lose those actins, so we're probably somewhere near the M line. So depending on where we cut through a sarcomere, we're going to see myosin or not. We're going to see actin or not. Everybody understand how we get these cross sections? Make sense? So I guess the other thing about our proteins, we've got a lot of water in those muscle cells. What do we know about those outside of the protein? Water is going to act as a lubricant to the outsides of those proteins. Those sarcomeres are probably going to be very hydrophilic. They're going to like water a whole lot. Water allows that motion to happen a lot easier. Discuss those. So that Z-disc, the Z-disc provides a certain amount of elasticity to the muscle, and that has to do with its structure. So each of these stumps here is where a thin filament attaches to, and you can see that there's a little bit of flexibility there. As there's tension on that, you can see things move it a little bit, acting as shock absorbers, at providing a little bit of flexibility, a little bit of elasticity as we connect one sarcomere to the other. So actin stumps, we've got that Z disc or Z band there in the middle. So as you pull out of the cell and look down on it, you can see our sarcomere again, Z line to Z line, your A band, your I band. We can add to the diagram where our M line is, where our H zone is, moving forward. So how many sarcomeres might be in one muscle fiber? This is not something I'm going to ask you to remember, but I thought it was interesting to do the calculations. If we assume that the muscle fiber is 3 centimeters long and each sarcomere is 3 micrometers long, do the math there, and there are 1,000 myofibrils in a particular muscle cell. Doing the math, figuring everything out, we're looking at 10 million sarcomeres in an individual muscle fiber. So thinking about our sarcomere, how much does it contract? What's the limit to contraction from what we've seen so far? What's going to slow things down? Or at least stop the contraction? Does anybody want to guess? Z line, they're coming together. Why don't they come all the way in? What happens? Ones in the middle might hit the Z disc first. So maybe limited by the length of the thick filament or the myosin complex, we can only come down to that much. Till we hit the ends of that, or when the actins hit each other. That's as far as we can go. We've overlapped the actin and myosin as far as they'll go. So we're looking at from 100% down to 60%. So why not less than 1.5? We've already said the thick filament's going to limit how far those Z discs can come in. Why don't we want to go 3.5? If those myosin and actins stop overlapping, it's going to be hard to line those back up again. So we want to always have a little bit of overlap there. If we don't, we're probably going to damage our muscle in some way, and then our lysosomes are going to have to come in, rip everything apart, and then reconstruct everything. So we want to avoid that if we can. So we know our parts. Let's get started on contracting the sarcomere. What's the process that allows that to happen? Muscles. So here's the picture here of our myoneural junction. We've got one nerve per muscle fiber. So you've got your muscle fiber here, cross section. We've got a lot of myofibrils in there. We've got a nucleus on the outside, part of our muscle fiber. We've got our neural junction, where the signal from the nerve comes down, excites the membrane, and that's going to cause the T-tubules in the sarcoplasmic reticulum to release calcium into the sarcoplasm. So muscle contraction starts with a nerve impulse. That could be from the brain, or you have reflexes where a signal may start in the spinal cord. We'll talk about them when we get to the nervous system. So that nerve impulse comes down. That nerve button, that nerve end is on the sarcolemma. The impulse is transferred across that gap. You saw that those two weren't exactly touching. There's a little tiny gap between there. So you have an electrical signal coming down. It throws a chemical signal out the end. That hits the sarcolemma. The sarcolemma then is excited and the signal is transferred to the T tubules, the sarcoplasmic reticulum, to release calcium. So nerve meets the muscle cell, as we mentioned already. We have one nerve for one muscle cell. So that's a wiring that's set by the time you're born. And all you can do is make your cells bigger or smaller. You can't change the number of muscle fibers or muscle cells. So that's why the number of muscle fibers is set at birth. You have that relationship between the nerves and the muscles. So at the end of that nerve cell, there's acetylcholine is released that goes across the gap and starts the release of calcium from the T tubules. That release of calcium starts a cascade that contracts our sarcomere. So we'll end there for today and continue on when we get up to the dairy next Thursday. Remember, we've got an exam Tuesday. Does anybody want to do any review or discussion right now? Does anybody want something on Sunday or Monday? I'll take that as a no. Okay. So 11 o'clock here. You'll have as much time as you need. So I guess there's a few of you, remind everybody that next Tuesday is our first exam. It's up on Blackboard, but make sure everybody knows that.
Can you post this lecture on Blackboard?
Yes. Okay, I'm going to try to work on getting all these things posted this afternoon.
Okay.
I'm not sure I'm going to get to it, but hopefully before the weekend, everything that related to this will be up, okay?
Thank you.
You're welcome.